Hello and welcome to the spring 2025 semester of Bioinformatics. In this class, we're going to learn a couple of ways that we can use computer science to solve problems in biology, but we're really just going to scratch the surface in this undergraduate course to look at a couple of really key applications. So looking at the microbiome data and understanding what microorganisms are in a community, including the human gut. We'll look at proteomics data and understand how to process that data and get key insights from it. And then lastly, we'll look at genomics data and specifically look at personal genomics to understand how variations are called and also how to interpret those variations. In order to get started in the class, the first group of assignments in our course are all tutorials. So they're going to introduce you to the tools that you'll use. Some of these are online websites like NCBI and Ensemble, where the information is stored for us to do projects and start to understand what kinds of problems we can solve by writing code to analyze biological data sets. Some of these are programming tools, so Posit Cloud or RStudio, either version of R that we want to use is going to be a really powerful tool in this class. And then lastly, Galaxy, which is an online portal that enables us to launch large applications that take a lot of memory on a computing cluster rather than running those applications on our own laptops. By the end of the course, you'll have a really good idea of some of the ways that you can use computer science for biology, and you should be able to feel comfortable with the different biological file types. So one of the first assignments, besides attending the Zoom that we had yesterday, is to make a little dictionary for yourself about the kinds of files that we'll use in this class. So you can, use all of these websites that are here. You can go to GitHub, you can go to Kaggle, you can go to public repositories that have a lot of data and find a version of each of these files that we're going to use in the class. And you'll be able to use this throughout the semester as a glossary to look up what you're working with. If you are really interested in this class and you really enjoy bioinformatics, one new option that we have at Endicott is that you can do a fifth-year master's in bioinformatics. So I'm also happy to answer questions about that, like what would come next once you have these quantitative skills. It is one version of data science. So I think one thing that's really great is that a lot of the toolkit that you use in R and Python is widely applicable beyond biology. And the master's in bioinformatics is designed so that you would start in May and you'd finish in May. So it really is a true fifth-year program with only 30 credits. 6 of those credits can be internship. So you can do all of your required courses, and then the spring semester you can do 2 elective courses, and then you can choose either a 6-credit internship or a 6-credit thesis to complete your degree. And if you want to find out what's happening in our area, I also recommend on LinkedIn joining the Boston Computational Biology Society, and I can send you an invite to this group. There's a lot of bioinformatics work that happens in Boston. A lot of the sequencing that you'll see on Ensemble and on NCBI has been done at the Broad Institute for large genomics projects. Even closer to our campus, the Gloucester Marine Genomics Institute is the place where a lot of ocean organisms have been sequenced and their full genomes have been assembled for the first time. So we're really in a hub of this work on our campus. And I think you guys will find a lot of uses for this class, both in bioinformatics and outside of it. Because once you have the toolkit for data science, you're going to start to see ways that you can apply reading in files, cleaning up data, and making beautiful plots that will help you in your thesis and capstone courses during senior year. Because this class is fully asynchronous, I will post videos, you'll get information from me, I'll have lots of announcements. But if you want help, just stop by my office or send me an email or set up a Zoom. I am here to help you. What I found is that it actually is more helpful to have one-on-one meetings with this material a lot of the things that you get stuck on are small syntax errors, and we can find those in one-on-one meetings a little bit faster than we could in the classroom environment. So I'm really excited to welcome you to this class. Let me know if you have any questions, and I hope that I get a chance to meet with each of you one-on-one before the end of the term. Have a great day.